Hello everyone, SMC here. Thank you for joining us today. In this video, I'm going to give just a brief introduction about SMC Laboratories and the services that we offer, including our model lineup. Let's go ahead and begin. SMC Laboratories is a Tokyo-based CRO specializing in inflammation and fibrosis. We were originally established in 2006 as a pharmaceutical company, but in 2015 our pharmaceutical hub was acquired and we became a specialized CRO. However, we believe our pharmaceutical background allows us to successfully aid our clients through the drug discovery process. Speaking of our clients, SMC is very happy to report that we have almost 800 clients worldwide from over 30 countries, and we have an almost 90% service repeat rate, with each client doing about 2 to 3 studies each. Here is a small example of some of the places our clients come from. Next, we have our disease model lineup, which I will now go over briefly. Our main organ of interest is the liver, as this is the organ we have the most experience with. For NASH studies, we have our proprietary Stam model, which is our most popular model, and shows a neat progression from steatosis to NASH to fibrosis to HCC. We also have the CDA high-fat diet feeding model. For the more severe cirrhosis models, we have the CCL4 induced model as well as the bile duct ligation BDL model. For the acute liver fibrosis models, we have the CCL4, DGAL, TAA, and the CON-A models. And finally, for hepatocellular carcinoma, or HCC, we have a proprietary Stam model, as we do see HCC occur in the later stages, and the DEN-CCL4. Next, we have our lung models. First, we have our bleomycin-induced idiopathic pulmonary fibrosis, IPF, which is our second most popular model in our lineup, as well as our chronic obstructive pulmonary disease, COPD, model. For the kidney, we have several models related to chronic kidney disease, including our unilateral ureteral obstruction, UUO, model, which is our third most popular model, as well as the adriamycin, adenine, and folic acid models. For the skin, we have the systemic sclerosis model, which comes in 2 types. The first looks at just the severe fibrosis of the skin, while the second kind looks at the progression of the skin fibrosis into the lungs. Finally, under others, we have our Alzheimer model, orthotopic xenograft model, as well as custom models. By custom models, we mean 2 things. 1, clients can bring us literature that features a model that they would like to work with us to recreate, or 2, it can mean changing one of our already existing models to fit the individual needs of the client. These are just some of the services that we are capable of here at SMC Laboratories. Finally, let's take a brief look at our client base. We have a very diverse client base, including companies with compounds already in clinical trials, megapharma and bioventures, as well as institutes and universities. No matter who the client is, though, we are always happy to broaden our client base and find ways to accommodate their needs. Thank you for watching our video today. After seeing this video, if you have any questions about the information that was presented here, or about how SMC can help you with your drug efficacy study, please do not hesitate to reach out and contact us. I will put our contact information and a link to our website in the description below. We would be happy to answer any questions you have. SMC Laboratories looks forward to hearing from you soon. Welcome back, everybody. Today I'm going to explain a little bit about some of the HCC trials that we have done using our proprietary Stam model. If you haven't already seen the Stam model introduction video, I would recommend watching that video before continuing on with this video. I'll go ahead and put the link in the description of this video. And with that, let's go ahead and take a look at our first slide. As you can see, we have done over 30 studies, both therapeutic and preventative, in the Stam model that target HCC. Down below, you will see a list of 15 presentations that are non-confidential and can be shared. Today, I will be going over these 3 slides with you. The first study that we are going to cover is a study that we did with Genentech. This was a TGF-beta neutralizing antibody called 1D11. It was given at 10 milligrams per kilogram IP twice a week. Administration began at week 17 and finished at week 23. We observed that this compound successfully decreased both the size and the number of nodules, as you can see in the first two charts here, as well as significantly reduced fibrosis. Finally, we observed that it had a significant effect on the T cell distribution, as you can see in the final chart. The next study that we are going to look at is from a study we did with Sempra. Their compound was an antibiotic that was given at 100 milligrams per kilogram orally once daily. Treatment began from week 4 and continued until week 20. This is actually one of the longest treatments that we have had experience with here at SMC so far. This compound was able to significantly decrease the NAFLD activity score starting at 10 milligrams per kilogram, as well as decrease the serious red positive area, or in other words, the collagen deposition. Finally, we saw a significant decrease in the number and size of tumor nodules present in the liver. The final study that we will look at today is a study that we did with Kurume University here in Japan. We tested the DPP-4 inhibitor called stagdoliptin, which is commonly used today as an anti-diabetic drug. This was given at 30 milligrams per kilogram orally once daily from week 8 until week 17. As you can see in the chart below here, it successfully decreased the number and volume of the tumors found within the liver. Finally, here's some gene sequencing data. I'm not going to go into depth about this data right now, but if you would be interested in learning more about these studies that we have done here at SMC or about our gene sequencing data, please do not hesitate to reach out to us. We would be happy to provide you with more information. And that will do it for our video today. Thank you for watching, and we hoped you liked what you saw. As I just said, if you'd like to learn more about what you just heard or about the services that are available here at SMC, I will put the link in the bio so that you can get in contact with us. Thank you again for watching. Hello everyone, SMC here. Thank you for joining us today. In this video, I am going to give just a brief introduction of SMC Laboratories' proprietary Stam model. Let's go ahead and take a look. To begin with, I will share a few facts about our model. The Stam model is the most used patent model in the field of NASH studies. In fact, as you can see in the diagram, 41.3% of all companies currently conducting clinical NASH studies have used our model. We also have 15 successful CTA packages, with companies entering into phase 2 in clinical trials and beyond since 2021. Along with that, Stam has been featured in 60 publications and 80 presentations. If you are interested, please feel free to seek them out or contact us for more information. The next image that you see here is a list of pathologies that are observable within our model. I will not go into too much depth about it here in this video, but if anything catches your eye, please contact us so that we can set up a time to discuss it in further detail. On the next slide, you will see a diagram of the Stam model, which reproduces the human time course of NASH. Starting from simple steatosis, progressing to NASH, fibrosis, and eventually hepatocellular carcinoma. The only difference from the human time course is its incidence rate, as it shows a 100% incidence rate at each step. We guarantee by 20 weeks that 100% of all surviving mice will develop HCC. In the lower panel, you can see how we create the Stam model. We use C57 black 6J wild-type mice and induce the disease through 2 hits. The first is a chemical insult just 2 days after birth via a low-dose injection of streptozotocin, which induces a diabetic background by wiping out nearly 2/3 of the mice beta cells. The second hit is continuous high-fat diet feeding from 4 weeks of age. Both hits are necessary to induce the NASH pathology. This model is so timely that we can actually pinpoint when the different stages of the disease progression will occur. As you can see here, at 5 weeks of age, the mice show fatty liver. NASH is evident at 7 weeks, and by 9 weeks, fibrosis is established in all mice. At 12 weeks, we see nodules in the liver, and at 16 weeks, hepatocellular carcinoma occurs. One of the advantages of this model is that due to its very punctual and stepwise disease progression, the treatment period can be tailored depending on the mechanism of the client's compound and the disease target. In this slide, you can see some of the treatment periods that we would typically recommend to our clients based on what their disease target is. For a steatosis targeting study, we would usually recommend a treatment period of week 5 to week 8. For a NASH targeting study, we would usually recommend a treatment period from week 6 to week 9. For a fibrosis targeting study, we have 2 general study designs. For preventive treatment, we usually recommend a treatment period of 6 to 9 weeks, and then we can keep the mice in-house until week 12. For therapeutic treatment, we usually recommend a treatment period of 9 to 12 weeks, as the fibrosis is already well established at 9 weeks. Finally, for HCC targeting studies, we have 2 general study designs. One is for preventive treatment, with a treatment period of 6 to 9 weeks, and then simply housing the mice until takedown at 20 weeks. The other is for therapeutic treatment, with a treatment period from 16 weeks to 20 weeks during the HCC phase, where cancer is already established. Of course, these are just our most standard treatment periods. Designs are very customizable, and we are always happy to discuss with our clients to find the best treatment period for their compounds. Next, I will be explaining some representative data that has been generated in this model. First, we will take a look at some macroscopic images and histological HE stained liver sections. If you look at the pictures here at the top of the slide, you can see the visual progression of the liver throughout the different stages. You will note that by week 20, the HCC-related nodules are evident. A key endpoint for this model is the NAFLD score, which is also used for clinical patients as well. This score is calculated by using HE staining to look at the cell structures within the liver. It is used to observe the progression of the steatosis, the ballooning degeneration, and inflammatory foci, all of which are characteristics of human NASH. Because our model induces this phenotype, we can use the NAFLD activity score as a primary endpoint in this model. At 6 weeks of age, Stam mice have a NAS of 4 and up, so they meet the criteria for NASH. Next, I will review some general parameters of the Stam mice, including the body weight, liver weight, and serum biochemistry. Stam mice have a lower body weight compared to normal mice, but show characteristic weight gain associated with NASH, as well as increased liver weight compared to normal mice. Due to the fact that the body weight is lower than the normal mice, we believe this model does not have an obesity background. You can also see high blood glucose and liver triglyceride levels, as well as mild elevation of ALT and AST. Here are some metabolic parameters. The main thing to notice on this slide is that from the non-fasting glucose levels, you can see that while the Stam mice do have lower glucose compared to the normal mice, they are still producing some glucose. This leads us to believe that the mice have a diabetic type 2 background as opposed to a diabetic type 1 background. In this next slide, you will see some lipid parameters, including the serum cholesterols and triglyceride levels. You will notice here that they are all higher compared to the normal mice. One of the analyses that we are able to do here at SMC Laboratories is cytokine analysis via ELISA test. In this slide, we evaluate the results of 2 cytokines, cytokeratin 18 at week 9 and serum AFP at week 20. CK18 is a biomarker for NASH, and serum AFP is a biomarker for HCC, and as you can see, both are significantly increased compared to the normal mice. Next, I will go over the histological analyses that are routinely done for this model. One stain that we often use is oil red stain to observe the high fat deposition in the steatosis phase at 6 weeks, which decreases with age as fibrosis progresses. This is similar to the human condition and is called burned out NASH. Below that shows the F480 staining, an immunostain for macrophages. You can see macrophage infiltration in zone 3, with levels increased from 6 weeks and onwards. On this next slide, we have serious red staining for the collagen deposition and ERTR7 immunostaining for fibroblasts. Looking at the second picture for the serious red stain, you can see that the chicken wire fibrosis is evident, which is commonly associated with NASH as opposed to the more severe bridging fibrosis of cirrhotic diseases. On this side, you can see some time course data relating to the progression of the macrophages, collagen, and fibroblasts. This data was quantified using the serious red, ERTR7, and F480 staining, and as you can see, they all show a significant increase compared to the normal mice. Another analysis we have here at SMC is the qPCR test. In this slide, you can see gene expression data for inflammation and fibrosis-related genes. MCP1, TNF-alpha, and IFN-gamma are gene expressions for inflammation, and TIMP1, collagen, and TGF-beta are for fibrosis. On this next slide, you can see another endpoint that we offer here at SMC, micro-CT scans. In general, this is not something that we do for every study. However, it can serve as useful supporting data for those who want to see tumor progression within the liver during the in-life portion. The last slide that I will discuss in this video is the survival curve of Stam mice. With no intervention, we see relatively low mortality all the way to week 9 through the steatosis and NASH phases. However, we begin to see about a 30% mortality rate by week 12 and an almost 50% mortality rate by week 20 for the fibrosis and HCC phases respectively. And that will wrap it up for the introduction of our proprietary Stam NASH-HCC mouse model. If you liked what you saw, please like and subscribe. If you find yourself with any questions or an interest in using our models, please do not hesitate to reach out. I will be putting our contact information along with a link to our website in the description below. Thank you for watching our video today, and we hope to hear from you soon. Welcome back, everybody. In this video, I am going to do some further explanation into our Stam model that I talked about in a previous video. I'm not going to review that here, so if you haven't seen that video, I would recommend watching it first. I will put the link in the bio so that you can watch that if you are interested. Specifically today, I'm going to explain about the positive control for our NASH fibrosis studies, which is telmisartan. Telmisartan has two mechanisms of action. One is an angiotensin receptor blocker. Angiotensin pathways play an important role in hepatic stellate cells, which regulates liver fibrosis and liver inflammation. Therefore, it shows anti-fibrotic and anti-inflammatory effects in this model. The other is PPAR agonist activity. PPAR is one of the metabolic modulators, which is the reason why telmisartan shows anti-steatosis or anti-lipid accumulation effects in this model. I will now briefly explain the results that we have had with telmisartan as our positive control. As you can see here, in this study, we began administering telmisartan 10 mg per kilogram orally once daily at week 6 until week 9 at sacrifice. Here you can see the general conditions and biochemistry, and you will notice that the telmisartan treated groups showed a significant reduction in liver triglyceride as well as a decreasing trend in serum ALT, our liver damage marker. Here are some histological results. From the HE staining, we can see a significant decrease in the NAFLD activity score, and from the serious red staining, we can see a significant reduction in the fibrosis area. Looking at the gene expression data, we can see that the genes TNF-alpha, MCP1, alpha SMA, and TIMP1, which are related to inflammation and fibrosis, are significantly decreased. These are just some examples of gene expressions that we are able to test for. We can conduct expression analysis for any genes as long as the specific primer is available. And that will conclude this video on our positive control telmisartan. If you are interested in what you saw today and would like to ask specific questions or would be interested in setting up a study with us, please do not hesitate to reach out to us. I will be putting a link to our website in the bio. From there, you can send us an email from the Contact tab. Thank you for watching our video.